Now, we all know what DNA stands for, right? It's deoxyribonucleic acid. The only way it's different from an RNA molecule is the sugar. It has a deoxyribose sugar instead of a ribose entity. And also it's a double-stranded structure. But in addition to that, what else do we need to know about the DNA? So as far as the molecular structure of DNA is concerned, it is two strands coming together to form one DNA molecule. I believe some of the standardized tests would also require you to calculate the amount of DNA or the molecules of DNA. We are not doing that in this chapter today, but there are chapters where you would be learning how to calculate that. But two strands of DNA coming together make one molecule of DNA. What is interesting about the DNA is it is made up of sugar, phosphate, and nitrogenous base. Now, most textbooks would show you the sugar and the phosphate as the pentagon and the circle, and as the backbone, as seen as red in here. And then it would show you the bases facing inwards, shown in blue, green, orange, and yellow in this image. And you would see the two strands running from 5' to 3', and the other strand running in the opposite direction, 3' to 5'. How do you know which end of the strand is a 5'? You need to look for a free phosphate group. If you see a free phosphate group, the circle over here, you can see a 5'. If there's a free hydroxyl group from the sugar, then that would be the 3' end. What is important is to note the bases that complementary base pair with each other. Those are basically coming from the Chargaff's rule. I'm kind of assuming everyone knows that. If not, this would be covered in very basic biology. You have A going with T and you have G going with C. And these dotted lines are your hydrogen bonds. So it's telling you that the G and C has 3 hydrogen bonds between them. A and 2 have 2 hydrogen bonds. So you have DNA, and then you have the final product, which is the protein. How does the information flow from a gene to protein? That is actually based on one of the 2 DNA strands, or the template strand. But if somebody was to ask you what this flow of genetic information within a biological system or a cell would be, you would use the term central dogma. So what is central dogma? Central dogma just explains the process by which instructions that are part of the DNA are converted into a functional product protein via RNA production. So the DNA to RNA conversion is called transcription. And then the RNA to protein conversion is called translation. What I want you to pay attention to is the fact that sometimes RNA can go back and convert itself into a DNA molecule. And it is prevalent and seen for virus particles. We're not going to cover that in this workshop today, but you should know that it could go in both directions. So you could have a DNA to RNA or RNA to DNA. But as far as the translation part or the component of the central dogma is concerned, that's always one way, unidirectional. So you have RNA to protein only. So let's move on to the next slide, which explains what happens during protein synthesis. So in this case, you have a DNA molecule, you have 2 strands. At a given point of time, the cell is going to pick only one out of the 2 DNA strands. For simplicity purposes, most textbooks would use the 3' to 5' strand and show that as the template strand. But you should know that at any point of time, the cell can decide which strand to pick. The reason why the textbooks use 3' to 5' is because then they can easily show the RNA synthesis during translation from 5' to 3' direction, because that always happens in that direction only. So it's easier for them to pick just the 3', 5' DNA strand as the template strand. But the process, this is the first component of the central dogma process, which is called transcription, which is mainly converting DNA, 2 strands, into one single strand, RNA. In this case, the kind of RNA that you would see is the mRNA, also known as the messenger RNA. Now, for a given gene, the template strand is always the same. However, between genes, the cell can alternate between the top or the bottom strand as the template strand of the DNA. And I'll discuss that later in a bit. But let's cover and see what transcription looks like in a eukaryotic cell. So in this image, you're seeing two strands of DNA, top, bottom. They're blue in color. The top one, if it's running 5' to 3' left to right, the bottom one would be running 3' to 5' left to right. Again, as I mentioned, for example purpose, they take the lowermost. That shouldn't mislead into thinking that that is always the DNA template strand. Cell could use the top one as well. But let's say for just this example, the lower one has been chosen as template strand. This is where the Chargaff's rule of base pairing comes along. The C would go with G, the T goes with A. And so in pink, you're going to see the mRNA strand, which is the 5', 3'. Now, a lot of people don't realize this, that when transcription is happening, it happens in a very, very systematic way. It doesn't just start anywhere on the DNA strand. Your RNA polymerase enzyme literally binds to the DNA at what we call the promoter end, and then it zips through the DNA, reads it, and then it stops at the terminator. So, but before we get into that, let's take a quick look at one of the questions. See, what kind of bonds link base pairs in DNA? The clue here is the 3 dots, if you remember from one of the images that we just saw. So if you remember what those 3 dots look like in your reference material, you would be able to answer this question correctly. That says hydrogen bonds, that is the correct answer. So by the way, just so that you know, this question was, all the questions on this presentation were actually picked from previous standardized tests. And again, it just sneaks up on you, you know, you read about this and then you close the textbook and you're like wondering, okay, what happened? And it just sneaks up on you like this and asking you what kind of questions. It basically expects you to notice that those 3 dotted lines in any of the figures in your reference material are the hydrogen bonds. And that's actually the standard nomenclature for hydrogen bonds.